Okay, here we are. We're going to start our class. Welcome back, everybody. And here we are. So we're going to get started here, and everything's rolling. And this is working. Okay, great. So, yep. And so here's where we're at. So we're reading chapter 18. Chapter 18 is about programmed cell death and apoptosis. We're going to start talking about it today. I haven't mentioned yet in this course that chapter 18 is also about stem cells. We've learned about stem cells, and we're going to stack our knowledge on top of what we'd already learned on stem cells. Then you're working on your term paper. Great. I can help you with that. I've already helped some students as you work away on that. Work in those two documents. That's what you're going to be graded on. Then there's a cell cycle drawing assignment. You're sending pics of that to me today. And then there's going to be a stem cell drawing assignment that'll be due a week from today. And I haven't had a chance to make the video yet, but I'll make it today, more likely tomorrow. And so look for that bright face pose, and it'll be just another example of a video you watch, and over part of it, there'll be an assignment to draw parts of it out. But there's significant learning to do, because I can basically deal with all of the stem cell biology in chapter 18 in this video. Okay? So there's that. And then, amazingly, our second exam is 2 weeks from today. And it will be on chapter 17, 18, and 19, and of course, everything that we covered in class. So chapter 18 is about stem cells and about programmed cell death. And then chapter 19, I think, is just called cancer. And it's kind of similar to the chapter you would imagine would be in our course if I said our whole course is subtitled The Molecular Mechanism of Cancer. It does a lot of adding to, it's the last chapter in the book, it does a lot of adding to what we've already learned about. That's where June and Foss show up. That's where CAR T-cell therapy shows up. And but the author deals with a lot of that stuff in a kind of a superficial way, like just one figure about CAR T-cell therapy. It's also where tamoxifen and a modern pharmaceutical oncology and cancer biology is really dealt with like that. So it's a good chapter, and it's a chapter probably that, you know, there's going to be more expectation of you being able to answer questions from chapter 19 that we didn't talk about. Because it talks about the biology of cancer, like the carcinoma, sarcoma, and so forth, like that, and lymphoma. You know, what are the different types of cancer, stuff like that. So again, bring questions to class. I always have my textbook and bring questions to class, because we're reading chapter 18 this week, chapter 19 next week, and there's really a good chance, and then we won't have more than that on the exam, but there's a really good chance that we'll get done with that stuff fine, and we'll add a couple more examples of things that add to our knowledge before the exam. The chapter we'll read after this, so after our second exam, we'll cover a couple new chapters. And the first one we'll cover is chapter 10, and chapter 10 is about the nucleus, and we've learned a lot about the nucleus. FOXO can't get in the nucleus, right, like that. So there are some examples that we can actually add if we have time before the second exam. We can add for the second exam where you won't need to look at chapter 10 yet, but we'll actually be kind of foreshadowing into chapter 10, even though you won't need to look at it, because it just takes FOXO and explains, well, why is FOXO not able to get into the nucleus? In a little bit more detail, a little bit more explanation, like that. And remember the idea that you don't have to go back and cover everything that we learned for the first exam, but there are features of the second exam that are really building on the first exam. For example, we learned about ALK, and phosphorylated ALK activates MYC. But for the second exam, we learned that phosphorylated ALK is binding together with SRF, serum response factor. So you might want to go back and just remember that from the first exam. I won't ask about RAS because we really didn't stack knowledge onto the RAS story for the second exam. Another thing to think about is the second exam's worked a little more than the first exam, you know, so that's kind of what we want is students get more comfortable, right? A lot of students want to do better on the second exam, and so they'll try. I think they'll be more comfortable and aware of the expectations, so, you know, add a little more credit for the second exam. And then, oh man, you know, this is, you know, spring semester where the final exam comes very soon after the second exam. So you're also starting to study for the final exam, you know, and it's one of these things where it's worth your time, you know, to kind of really be pulling it together now, so that you'll be, you know, getting points for the final exam, because the final exam covers everything. And we'll cover a couple more chapters, and there'll be new material on the final exam, but it just covers everything. So it will cover everything that we have. And you know, the whole course is taught with a big, big goal of having stuff that you can remember, because it's just a framework of these giant fields of molecular biology and cellular biology. It's just a framework for these, and you really need to hang on to that structural framework to be able to be like, oh, okay, I remember that. You know, oh, it's a transcription factor. Yeah, like last time, right? Mallory mentioned, well, she had heard about P53, knew something about it. Well, you know, here's something you will never forget. P53 is a transcription factor. That's its job. It's the guardian of the genome, and how does it do its job? It activates target gene expression. It's really the target genes that are the effectors, the role players. And P53 is just a regulator of this, right? And we learned that P53 can activate P21, and P21 is just a small inhibitor of the start, and it's just like, don't go. Don't go. There's DNA damage that we need to correct before we dive into S phase. Yep. So that's that. So that's where we're at, and of course we're rolling now, right? We're rolling with this mechanism of our course. And so do people have questions about anything at all they want to ask right now? Yep.
I have a question about the term paper. Is that?
Yep.
Okay.
Yes, yeah. So in general, you're working on your term paper. You know, it's each of you pick your own topic. So you're working on it your own. But there's the video, and then there's the syllabus that describes what to do on the term paper. But you're welcome, it's super welcome to ask questions, and then my job is to turn it into a general answer about term paper.
Okay. Is it okay if we copy, so like I've been writing things on my notes, and then I copied and pasted my notes into the writing to then build off of that? I just know you said you didn't want to see a bunch of copying and pasting in the writing, but is it okay if we copy and paste what we write into?
Yeah,
Okay.
so yeah, there's a good chance you haven't written a term paper like this before. You have two documents, and one is notes and one is the paper. And the idea is you're documenting your work in the notes. And so that's going to be messy because you don't need to delete anything. Just keep pasting stuff in there. More stuff you paste in, the better. And especially if it's over time. And it shouldn't be big chunks of stuff. But it can be, as long as those chunks don't then just get pasted into the paper. Because what I want to discourage is broad use of AI. Small use of AI is great, right? Google does that now for you, right? I post that up here a lot, AI overview, right? And it's just like, whoa, that's nice writing, right? You're welcome to use that. Paste that in, use it as a source. You know, just use it as a source, right? Because you can use any source. You're welcome to use AI as a source, any source of AI. You just have to include it in the bibliography as a source where you use it. And it shouldn't be like you shouldn't have a blank document for notes and a one-time paste of a full paper into the paper document and one item in your bibliography that says just AI, right? That's right, because I won't call you out on that. I won't say like, oh yeah, that's an F and a plagiarism and a false use of you know resources. I'll just be like, yeah, well, I'm not impressed with that, you know, because it's like, it looks like you just pasted that in and basically you're admitting it, you know, like that, right? So that's the whole point. And again, if you're anxious about that idea, just try to do as much as you can over time in the document, because I can see the editing and stuff like that. And I'm not, you know, I'm not like a vicious, mean-spirited person. You know, I'll be like, this looks good. You know, I just want to show more of your work over time, so that I can be like, yeah, you know, this is good writing, and I guess you're a good writer. You know, because you know, in the past I would just be like, wow, you're a good writer. Yeah. There was a student that took a course once, and I was like, this is such good writing, right? It was a long time ago, long before AI. It was such good writing. And that student became like a science writer and then got a free scholarship to Northeastern Law School. Yeah, just shocking, you know, like that, because wanted to write and then, you know, communicate about science, law stuff, yeah, you know. But at the time, I just shrugged because it was just such good writing. I just shrugged and I'm like, that students don't write like that, right? It was such good writing. But, you know, I talked to the student, not challenging them, but just being like, wow, that seems like really good writing. And I asked them about how that happened. And they said, and I wasn't, challenging them and saying, I'm going to charge you with plagiarism. I was just curious, because it was surprising. So yes, yeah, so the notes and the paper. And the paper should have editing in it too, right? You're writing, right? Like that. So, and it's probably a new experience, but it's really what I hope a student would always do, right? Is they're organizing their notes somewhere. And remember I said some students really like to write in their handwriting. Well, you could paste pictures of that in, right? That's really convincing that that's your work, right? And, and of course, like, if you want to know something, well, I won't tell you something, but of course there are workarounds with this, right? Students can find any way now they can ask AI, here's the rules for this paper, how can I cheat, right? Literally, yeah? Yeah. But I should tell you that I have a daughter who's an English teacher in a really high-end private high school in Manhattan, and I'll talk to her about that too, because she knows all the stacked up mechanisms of how AI can help these punk-rich high school students. And is this going out on YouTube? I withdraw that statement. I withdraw that statement. And I love those kids down at that school. They're just doing great and they're working hard and doing their job. All right, so yes, that. And I could talk to you individually about it if you're worried, or how do you do that? Yes. Okay, other questions people have? Oh my gosh. I refuse to edit any of these, but I probably should.
Just take it out.
Yes. Right? Yeah. Other questions people have about anything at all? All right. So just interrupt. When you're thinking about something, just interrupt anything, and it's my job. So again, we've heard from Sam. That's going to be part of our exam, of course, and part of our final, of course, is pulling it together. And we'll hear from him one more time when we get after the second exam, and we're I think it's after the second exam, right? Yeah. Oh, we didn't quite schedule that. Yeah, so we'll do it after the second exam, and we'll talk about that. And that'll be more about just the kind of the clinical experience and success of this treatment, right? And so, again, kind of what you would think, if somebody's going to talk about CAR T cell therapy, they're going to talk about the patients and how successful this is, like that. Yep. So we'll hear about that again, but after the second exam. And now, hopefully you've heard and seen announcements of this internship. And the internship is always about these kind of topics. That's molecular genetics of mice as a model organism to study human health and disease. And it's just a fantastic internship. And I mentioned that Joseph was down there, Sam is down there right now, and others of you, if you're going to be around next year, you should apply for this, because it's the best internship experience that I've ever seen. And we have so much success. Just this morning, there's an email now, USM Marketing has gotten permission to go down there and film a student doing their internship down there. And because it's just an incredibly fancy, state-of-the-art research facility. And USM is like, we would like to just piggyback on that, and convince students to come here. And it's a good idea, because it's a good part of college education. So, and I'm happy to talk about that specific opportunity or any other opportunity to help strengthen your college degree. Great. All right, so we're talking about the cell cycle, and now we have this situation where it's actually the last piece of this chapter that I want to talk about. It's this P53, because P53 is involved in a DNA damage checkpoint. And we have two DNA damage checkpoints that we're learning about, and they're very different. So that is a great question to ask about in the second exam. The first one we learned about is this nice package over here. That's the G2 to M DNA damage checkpoint, and that involves CDK1 and cyclin B and the inhibitory phosphates and CDC25 and CHEK1 and that pathway. Very simple circuit to recognize DNA damage and stop. Stop what? Stop the entry into M phase. Now, we're over here, and we're at start, and we like to call this a control point, because it's a big show of whether the cell decides from external forces, signals, if it's going to go through the cell cycle. Included in it is a checkpoint, and it's this P53 damage controlled checkpoint. And P53 is going to be regulated and stabilize in the presence of DNA damage and get to do its job. And its job includes binding to P21 gene and activating expression of P21, which is a small inhibitory protein. So one reason why we wanted to really say, no, we're way over here. We're on the other side of the cell cycle. Totally different point, totally different story, same concept, DNA damage. You better stop and wait, okay? And so we're at this regulation of start, and we're just focusing on this DNA damage point, okay? So it's the guardian of the genome, woohoo! And it's like that, there's that analogy, and it's a tumor suppressor, and we learned about that. And then it's in our story here where we focus on examples of start regulation, okay? And so this last part down here, it's the DNA damage checkpoint. This is all about turning start on and turning the light green. This is the school patrol. It's an independent story, and it's like, I can make the whole thing stop. Even if the light is green, I can make you stop, okay? And our book shows this, and it shows that in the absence of DNA damage, p53 is just continuously destroyed. And then in the presence of DNA damage, a different check, kinase check 2, phosphorylates p53, stabilizes it, and when it's phosphorylated, it joins our list of proteins that are transcription factors that are activated when they're phosphorylated. So it's like ELK and STATs and SMADs and more. Gets turned on like that. And it just binds to P21. It has other target genes it binds to, but this is a really sweet one where you're like, got it, memorable. It activates a small inhibitory protein, and P21 just says stop. Says, I'm school patrol, boom. You have to stop, and you have to wait as long as the P21 is out in the intersection with the flag. Wait. And the cars start honking, the cells start honking, they're like, we want to go, we want to go. And they're just like, nope, not until that goes, okay? So here's the last part of it, and that is, as the DNA damage is being repaired and the cell is waiting, this check 2 just keeps phosphorylating p53 on more and more sites. So initially, it just phosphorylated on one site and inactivated. Now, it's going to hyperphosphorylate p53. And it's like a little timer mechanism where it's like, I'm guarding the genome and there's damage, and I'm going to give you a chance to correct it. We're going to wait, we're going to wait. And as we wait, I'm just going to continue to phosphorylate p53. And when p53 gets hyperphosphorylated, it's going to have new target genes. So that's the first time we've seen that. It's very unusual that a phosphorylated protein can get hyperphosphorylated and somewhat change its activity. And when it gets hyperphosphorylated, it binds to genes that we don't need to know the name of, that we're going to learn about in chapter 18, and we don't need to know the name of then. We'll get to that today. And those cells are going to kill, those genes are going to kill the cell. So that's a big p53 job, is to say, wait, and as we wait, I'm getting tired of waiting. And if you can't fix this, you should go through apoptosis. Yep, it's just a timer mechanism. And it's as simple as that, right? When we get down to the genetics and biochemistry of molecular biology, the mechanisms are like that. Like Mallory said, had heard something about p53, right? That's in the newspaper and TV shows, right? Like that, it's the guardian of the genome. Woo, here's how it guards it, right? It's a transcription factor. Most transcription factors only do that. They only are transcription factors. They don't go off and do something else. So here's a good example of that, where it's a transcription factor and it just shifts its role, right? From waiting to killing, right? To saying, no, tired of waiting. And so it's just a built-in timing mechanism. Nice. That's it. And that's our last piece from chapter 17, and it's a foreshadowing into chapter 18, because you see these funny names up here, and don't worry if you can't read them in the back, because we're not learning the specific names. We'll talk about them. But they are going to stimulate apoptosis, and apoptosis is cell death. The cell is just going to die. Yep. Okay? All right, so that's that. And the book has nice figures. This one is actually ahead in the cancer biology chapter, because the book wants to loop back to some of these examples. and show that idea, because there can be mutations in this system that contribute to cancer. Remember that p53 is a tumor suppressor. So I just added those, and then we'll roll on to the next chapter. And so we're fitting that all together. Oh, let me just scroll back to showing you a couple of kind of summary slides we started with. And so remember, the parts of chapter 17 we focused on is that we went over to the G2 to M transition and the DNA damage checkpoint. Then we learned about the regulation of start, and we have those examples that we'll look at too. We have kind of 5 categories of examples. And then both in class a little bit and in the cell cycle video talked some about the phases of mitosis, and that's in your book too? Yeah. And so in regard to the regulation of start, we have this summary slide. We said 5 examples of start regulation, okay, to kind of categorize that. And our book talks about these, and there's a way to walk through it. We added to transcription regulation that's really focused on the cyclin genes at start. They can be rate limiting, we just need more cyclin. That's what's happening on the skin cells under a scab. They're just waiting at start, they need more cyclin. And then retinoblastoma, the first tumor suppressor. It's a transcription factor, it's a co-regulator, it's a co-suppressor of E2F. And E2F is just, it's a family of proteins, and it just has an amazing list of target genes. Because E2F says, when we go through start, what that means genetically and biochemically is we ramp up synthesis of DNA. We have a lot of genes we need to either activate or increase their expression. A good example like that, what would you list some target genes of E2F? You can just guess at them. Well, probably a lot more DNA polymerase. That's an enzyme, that's a protein, it must have a gene. Probably need to make a lot of that in S phase. S phase is a huge job. The cell can't do anything else. Then what does DNA polymerase need? Well, it needs a lot of helper proteins. And you might remember from intro bio that it needs helicases, topoisomerases, and so forth to get the job done, right? Then it needs the building blocks. Those are biochemical products of metabolic pathways. We're just gonna ramp all these up. Enzymes, enzymes, enzymes to synthesize more. What about DNA repair? Yeah, we've been trying to monitor that all along, all the time, monitoring DNA repair. But when you're synthesizing the entire genome, probably gonna be a lot of DNA repair. So we're gonna ramp all that stuff up. And E2Fs are big players. There are other ones too, but E2Fs are big targets. And so that's a big show. And then retinoblastoma is just like, nope, 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 nope. You wanna go, you wanna go, you wanna go, nope, 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 like that. So it's an example of one of these things that becomes kind of an Achilles heel in the plant. Lots of checks and balances, but that's kind of a vulnerable spot. Because if you mutate it randomly, you'll probably hurt it, probably damage it. And when it's damaged, its tumor suppressor activity can go down. Because the cell just leans into start, and it leans into start going, I don't care if there's DNA damage. I see that P21 out there, but I bet we can get around that checkpoint somehow, you know, like that. So it's a really good example. Then that P27 was just that isolated example, specifically to respond to Sam's well-timed question about how do you get this big switch? How does it turn into a big switch? All the checks and balances have to fall away, and the light is red, and then it's kind of flickering green, and then it's like green, 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 green. It's getting bigger and bigger, like that. And so that's just a good example to be aware of that there are all these feedback loops. Some are negative feedback loops trying to keep the light green, red, you know, and others are like positive feedback loops. When you want to go, like that. And so then there's that one, which is the actual DNA damage checkpoint of regulated start, you know, having that school patrol way of you know, blocking, no matter what else, you know, we're going to stop, okay? So again, that's the organization, that's the flow through the chapter. One thing I'm always trying to do in class is model when a biologist reads books and papers, they're trying to organize it like this in their mind. A classic question for any scientist is, well, what's the take-home message here? You know, right? You'll hear like some seminar and the scientist is talking about 50 different gene stories, and you're like, well, what's the take-home message here? You know, and they're like, good question, you know, because they have to say, no, that's a good question. I should settle down and try to give you the memorable pieces that you can hang on to, and then when you hear more, you can be like, I'm building on that strong foundation. So again, as I scroll toward chapter 18, let me know about questions. All right. So, and I was realizing that for the students in the lab class, they don't ask enough questions. And I was thinking about that because students in lab class don't ever ask questions enough. And I realized, just today, I've taught for a million years, and just today I was realizing in lab class, it seems like the questions are different. Because in lab class, they're questions of clarification, because it's about an activity. And so the questions are like when you get a job at Walmart, and you're working, and the manager says, I need you on the front end to run the cash register. And you say, how do you run the cash register? Right? They're questions of simple clarification. They're not fancy questions, like how do you build that cash register, right? And for me, it seems like I was realizing today that lab classes are the better setting to try to help students kind of start getting in the habit of asking questions, right? And I try to do that. I try to build the lab class so students will be, forced and encouraged to ask questions. Because that seems like an easier setting. Here it seems like in lecture class, this question should be kind of sophisticated. Like, well, how do you build a cash register, Walmart manager? And they're like, you're fired, right? But they just questions of clarification. And, so you can have your book and you'll be like, well, what do you have to know about this table? And I'll be like, nothing. There are many times in college when I would show the professor after exam, I was like, that was a good exam, it's pretty hard. I was surprised you didn't have any questions about this chapter in the book. And they're like, yeah, it wasn't important. And I'm like, oh my God, I wish I would have asked that question before I studied for the exam. There's a lot of time when you follow up with an instructor and you ask about that on the exam, and they just shrug, and they're like, yeah, no, we didn't even talk about it. And you're like, it's in the book. And you're like, yeah, no. Yeah, so it's really good to ask clarifying questions. Just clarifying questions like that, I always say this, I think I probably mention this every time. I hope I do. In college, I would just ask questions. And I got booed. I got booed 3 different times by my classmates where they're just, shut up, Champlin. Because it seemed like I was just way behind, like mind-bogglingly far behind. And side note, in my senior year in college in Minnesota, snowy, cold Minnesota, I went barefoot all year. So maybe it was a combination of booing that and my just apparent loser status. I also had no dates in college. Think about that. Just none. Yeah, at all. Nothing close to a date. All right, so chapter 18, program cell death. Why do people call it program cell death? PCD, because there are 2 very different, very important mechanisms in cells, eukaryotic cells, for program cell death, PCD. We're going to talk primarily about apoptosis. Apoptosis is what's going to happen in that CAR T cell therapy. The CAR T cell is going to kill itself. And so we'll talk a lot about that. The other kind is called autophagy, and autophagy is a super fascinating story. It's dealt with in our book in a chapter that we're just not going to get to in our course. So they're different. And we can kind of talk about, and here's the basic difference. Oh, so program cell death is a big one, and then stem cell biology is the other topic. It's actually the first topic in our chapter 18 is like half stem cell biology and then half program cell death. And we're going to go right to program cell death because we just talked about P53. And also because I'll make this video about stem cell biology and get that covered. We have a good foundation to talk about stem cell biology already. So autophagy and apoptosis. And you can distinguish them by drawing the cell. And so here's a cell that's going to die. And for apoptosis, what's going to happen is that the cell is going to die, so this cell dies. And we should say here, just to clarify this in this situation, the cell can die because of some internal signal, like that we just learned about DNA damage. So there could be an internal signal, like DNA damage, etc. Lots of things can go wrong inside a cell, and it's like, I'm out of here. I'm just not having a good day, right? And alternatively, and actually someone asked a question about this, I think maybe when Sam was talking about stem cell, CAR T stem cell, is there could be some external signal that gets it started. It's an external signal, like through signaling factors and receptors, but ultimately the cell dies of its own choice. The cell dies and it chooses to die. And there are specific things that happen. There's sort of like hallmark features of apoptosis that happen. Here's the key feature that distinguishes it from cell death for us is, the other kind of cell death for us is that there's going to be another cell that's going to phagocytize the dying / dead cell. And they're going to recycle the parts. And our immune system does that a lot. Is a cell's dying, it recognizes that. We'll talk about that in just a few minutes. And so this is the kind of the simple drawing story of apoptosis. Either an internal, external signal tells a cell you should die, and it does, and then another cell goes gulp, engulfs it and breaks it all down. And we want to hang on to all those amino acids and those nucleotides and sugars, and recycle them like that. So that's going to happen in that cell. So here's autophagy. And so autophagy is a different story. And at this level, what you'd say is it looks like this. So we'll just draw it up, a little thing like that. And in autophagy, the way to draw it is, and this is accurate, is the cell phagocytizes itself from inside. There's not an external cell that's going to do the recycling. It's just the cell itself that's going to recycle. And there's an organelle that just appears. The components are ready, and then it just appears. And so again, you don't have to read the part of the book. Just from lecture, we'll learn just a little bit about autophagy in our course. And the organelle is called an autophagosome. And it literally just phagocytizes things inside. And autophagy can involve, autophagy can occur during, we'll call it malnutrition or starvation. So it's very, very common for autophagy to not kill the cell, but just consume big parts of it. Because the autophagosome can engulf mitochondria and other organelles, engulf like a bunch of ribosomes, and say, ribosomes are made of proteins and we need more amino acids just to get through our day. Even if you just fast, you turn on a lot of autophagy. The cell lives, but it's recycling. It's like we gotta change our priorities around. And autophagy is super common in nature, like that. When like leaves turn brown, or like insects go through metamorphosis, the cell is just going like, I'm going to start recycling these components, and I'm going to start sending them back, to the plant to save, this, and I'm just going to slowly wipe myself out. Because if it gets extreme, the cell is just going to die. And really, it's just it doesn't have anything really necessarily to phagocytize because it's just chewed up itself. There's still some cleanup that needs done. But it's been recycling and sending away these nutrients and ingredients all along. And it's just like, I'm just going to just keep going because we're in really big trouble. Yep, like that. When somebody doesn't exercise and their muscles atrophy, my understanding is there's quite a bit of autophagy there, where muscle fibers decrease in size. And the autophagosome is just chewing up actual, the contractile fibers in the muscle. Lots of it happens in nature. It's a huge, huge thing. But scientists didn't understand it at all until relatively recently. So if you've learned about programmed cell death in classes, you almost certainly have just heard it referred to as apoptosis. Because scientists have learned about that longer, known about it longer, and it's a nice, solid story. And so we're going to focus on apoptosis because that fits in more in cancer biology. Our bodies are making cancer cells every day of our lives, and our immune system is recognizing them in a natural way and causing them to die by apoptosis, just wiping those bad cells out. And a lot of times cancer cells are just on their own, identifying an internal problem, like a p53 DNA damage checkpoint, and they just wipe themselves off, and immune system cell phagocytize them and recycles their ingredients. But our bodies are also doing this autophagy every day, remodeling, just restructuring what's happening inside of cell. It's remarkable because you're like, whoa, it's just gobbling up those mitochondria, and those are big, complicated organelles that take a lot of energy to build. And so it's remarkable, and it's kind of like that where both of these processes seem really contrary to the kind of efficiency of evolution and nature, where you're like, why did you make something that you're not going to use anymore? Like as fetuses, a third of our brain cells get wiped out because they don't make the correct connections, and they get wiped out by programmed cell death in a natural way. And you're like, dang it, I would have such a great GPA if I just had skipped that part of fetal development, But it turns out it doesn't work that way. So 2 big stories, and they're fabulously cool stories. We're going to move on. We're just going to talk about apoptosis. So does anybody have any questions about that? Okay? So especially think about this. For example, that thing, how does it know that that cell is in trouble and is in a dying state? Because it's going to eat it, whether it's dying or dead. It's got a plan, and it sees that, and it's like, oh, yeah, good, right? Okay? So that's what we're going to learn about, is that pathway. So here's just a little image of autophagy. Again, the autophagosome, it just appears. It's made of component parts that are always there, and it just assembles, and then it goes gulp, and it just turns into a lysosome, and it just breaks it all down. It's like a combination of a mouth and a stomach inside the cell, and it goes like that. Super cool. Tattoo? Yes, please, right? Right? We as a class, come on, you guys. The pathway, right, of the emergence of the autophagosome. So somebody's just going to have a little ink, because just little scattered little pieces, and then, we would be on the national news, you guys. Come on. And whatever the tattoo artist would be, they would start tattooing rock stars after our tattoos. No doubt about it. But we got to move on. Because here's the deal with apoptosis. And the story is that there are these steps in apoptosis that all eukaryotic cells do to go through apoptosis. And they are that there's a phospholipid bilayer around the cell. And there's a phospholipid bilayer, 2 layers. Let's just draw this out here so you can see this. All right, so here's a eukaryotic cell. I think this is probably, apoptosis has got to be restricted to, like plants and animals, multicellular animals, but definitely apoptosis in plants and animals. So very ancient strategy. Here's the phospholipid bilayer. It looks like that, We already learned from that phosphatidylinositol, that HIP3, that so when we think of lipids, we think of metabolism. Lipids have a lot of calories. Then we think of structure. Lipids can form structures. And then we think of information. So here's a combination of structure and information, because this is referred to as the outer leaflet. And the inner leaflet. And these are phospholipids. They're held together by non-covalent interactions And they are just hydrophobic interactions. It's very loose. The cell membrane is like soft butter. If you look, if you like tag a molecule here in the outer leaflet, and you ask, just by random diffusion, how long does it take to get onto the opposite side of a eukaryotic cell? For most of the phospholipids in the membrane, a few seconds. We can kind of draw and imagine the small scale of biology, but we can't imagine the speed of biological processes like just random diffusion. There are some special islands of phospholipids that are tied together because they're playing a bigger island-like structural, functional role. But for a lot of the phospholipids, they're just dancing around, okay? So they move in the outer leaflet just rapidly all around the cell. How often does that phospholipid flip to the inner leaflet? And the answer is never. Never, ever, ever on its own. Because to do that, you would have to dive the very hydrophilic head through the very hydrophobic lipid bilayer. And it just never, ever happens. Ever, ever. Just doesn't happen. And so, here's what the story is for apoptosis. Right now, inside of every one of our cells, in their inner leaflet are some of the phospholipids are, and we have to learn the name of this one, phosphatidylserine. Phosphatidylserine. And they're on the inner leaflet of all our cells all the time. And they're floating around, they stay in the inner leaflet. They were synthesized and placed in their inner leaflet by the smooth endoplasmic reticulum, and that's where they reside. How many phosphatidylserines on the outside of the outer leaflet? None. No worries, they're not going there, right? So what happens is that we'll meet a protein, because there are proteins in the cell membrane and other phospholipid membranes, and it has a specific name we'll learn, and in general they have a suite name, their general name, and some of their specific names, so it's a little bit confusing, are flipases, because they can flip specific phospholipids from one leaflet to the other. And there's a lot of that, and it's a big story, like some specific phospholipids get synthesized initially on the inner leaflet only, but then there's just a natural flipase process to get them to the outer leaflet, and vice versa, like that, okay? So we're gonna learn about a flipase that when a cell's going through apoptosis, this particular protein gets activated, and it flips the phosphatidylserines to the outside, and that's the target for that phagocytic cell. And the signal is, I see phosphatidyl on your surface, phosphatidylserine on your surface, you're dying, I'm gonna eat you. I don't care if you're in the process of dying or dead, you're just out of here, right? So on our list, one thing that happens on the apoptotic pathway is to flip the phosphatidylserine. Just flip some of the molecules to the outer leaflet, and that's a signal. That cell just doesn't stand a chance. Yep, okay. Does that happen in autophagy? Oh, no, nope. Right? So one thing is we know we want to distinguish between autophagy and apoptosis a little bit, so we learned a little bit about it. It's not like that in autophagy. There's no other cell involved. The autophagy thing is happening inside the cell. This is a thing where apoptosis features the phagocytosis of the dying cell. Yep.
Does that attract the cell that's going to engulf it?
100%. That is the signal. That is the attraction. That is the target. It's just that. That's how the phagocytic cell knows that. Yep. Okay. And that's fine. The cell is dying. That's just that. Yep.
So would also detected by hydrophobic and hydrophilic
Oh, I think so, well, so I think you're asking like, well, how does it detect it? Like,
Yeah.
so it has a receptor on the surface that specifically binds to phosphatidylserine. And normally that receptor is not activated, right? So it's gonna be like, it's gonna be a thing where it's just waiting, this phagocytic cell is just waiting with that receptor and others on its surface, but just waiting with that receptor. And if they start bumping into phosphatidylserine, it's like, here we go. Yep. So a cellular response, right? So we could back up and we'd say, well, I wonder what that signal transduction pathway is in that receptor. I bet there are some checks and balances on that, right? There's an amplification, right, signal and integration and things like that, right? And we don't know, it could easily be another pathway that we didn't learn about, but there's a really good chance that it's a receptor tyrosine kinase or a G protein coupled receptor that gets activated, and it's just like go, go, go. Yep.
Would there be an example where it's mutated or different, so it wouldn't be able to detect it, so that cell would just be living inside?
Yep. Yeah. And let me just mention a little bit about that. So for years and years, this apoptosis was a point of great debate in biology. So like back in the 1960s, they would stain cells under a microscope and they'd say, this is weird because this is healthy tissue, like a healthy fetal brain, not from a human, but like from a mouse. And look at all these cells dying kind of at the same time during what seems like natural fetal development. And other scientists would be like, yeah, no, they're not dying, because that would be stupid. Why would they be born and just die? Right? And it's important to appreciate that apoptosis is not death from old age or like starvation or injury. It's a specific type of it could be a relatively healthy cell going, nope, I got a problem, right? Like that P53 activation of these death factors, there would just be big fights. Like at science meetings, they would fight in the parking lot. They'd be like, no, I don't think so, right? Yeah. And so it was in the 1990s where some geneticists studying a little nematode worm, where they could point to every one of the cells. They could point to a cell, they could name it, and they say, want to see the cell die? Watch. There, it's gone. And so they started with a genetic strategy, and they found examples like you asked about, where there would be specific cells that would fail to die, and then there would be mutations where none of the cells died. So it was in the regulatory system, like perhaps in the receptor for the phosphatidylserine, where you're just like, you're not going to have any programmed cell death in this mutant organism. Yep. Yeah. And Sam, you have a question too?
Do you care about flat bases or scramblases?
So that's where, these are these flip bases have lots of different names and specific ones, and we're going to learn that one. So we're going to have a list, and we'll just learn that one. And it's a little bit of problematic with these because the names are used kind of both casually and specifically. Like that. So some of them are called a specific, it's a flip base, right? It's the phosphatidylserine flip base or the phosphatidylserine scramblase, like that. And so for our course, we'll say there are a bunch of them, and their job is to flip a specific phospholipid from one leaflet to the other, and then we'll just focus on the one then involved in flipping phosphatidylserine to the outer leaflet. Yep, okay. Because we're going to make a little list of the features. We're going to work on a list of the features of apoptosis right now, because there's going to be some activation inside to activate that protein and other proteins, like that. Yep, okay. So that's there. That's what the figure is showing there, is flipping the phosphatidylserine, and here comes the phagocytic cell. And that's the entire. There may be other examples, additional examples that could simulate it, but that's a big, big feature, is we're going to just flip the phosphatidylserine out, and I'm out of here. Right? So the book shows this figure, and what I circled is that these are sort of the actions that we're going to learn about, like the actions of apoptosis. And the first one is this one, translocation of phosphotylserine to the cell surface. So it just means to flip it from the inner leaflet to the outer leaflet. And this one is a protein that has both it's a problematic thing because it's kind of general terminology and specific, but we're gonna learn the term scramblase. It fits into this big category of flip bases, and the nomenclature of it is just kind of frustrating. But for us, we'll just learn scramblase. Scramblase, a scramblase flips phosphotylserine to the outside. And we talked about that. What we're gonna talk about with the red arrow is who does that? Who activates that scramblase? So we're gonna talk about that. But let's just look at the action. So there's a variety of different things that are gonna happen when the programmed cell death is triggered. Again, either by an internal factor like DNA damage, or an external factor, like another cell saying, you know, you should die. You look like a cancer cell. So here's the second one, just to mention, and that is that another feature of apoptosis that scientists saw for years is that what happens as the cell is dying is it just starts chewing up its own DNA. And I think there's a figure in your book that shows this. And this is satisfying for students because you're like, oh, this is a picture of gel electrophoresis. And what it's showing here is if you just put the DNA from a healthy cell into a gel electrophoresis experiment, that DNA is really long. Each chromosome is just one super long piece of DNA. And so that DNA is so long it just barely runs into the gel. When a cell is in the process of apoptosis, you see the DNA starts getting broken down. And you can see this way that it gets broken down. It doesn't just become a random smear. You can see, and people call that a ladder. So it's like this stepped series of pieces. And what's happening there that we'll be satisfied to see is that there's an enzyme, and it's a deoxyribonuclease, a DNase. We should write this down. So we've learned a little bit about DNases in our course. And I don't remember too much if we learned about them as much in our lecture or lab course, but here's an enzyme. And it's a deoxyribonuclease. It's going to chew up DNase, and people abbreviate that DNase. It's an enzyme that's going to chew up double-stranded DNA like that. And there are two kinds of enzymes, just to make sure I get this. So one kind of DNase bites the DNA right in the middle, just anywhere, just chew, chew, chew. And it's like a person that well, I don't know, it's probably not a very good example, but yeah, it's a person who bites spaghetti right in the middle. I guess we probably all do that, so it's a great example. This kind of DNase is called an endonuclease. And an endonuclease is involved in apoptosis. The other kind of DNase bites at the end. It chews in from the end, and that's called an axonuclease. And in apoptosis, we're going to activate an endonuclease. I drew the DNA like just as a DNA molecule, but we know the DNA is always wrapped around these nucleosomes. These nucleosomes protect. They protect the DNA some from that cutting, that endonuclease cutting. And they cover, each one covers, about 160 base pairs of DNA. So in a cell that's going through apoptosis, it's cutting up its own DNA. So it's definitely killing itself, right? It's going to get chewed up, phagocytized by another cell. But it's like, I don't need this DNA anymore. And if you think about it, that's a brutal strategy to just be like, I'll just cut this up. As it starts to cut, the first cuts tend to be between the nucleosomes. That's where the DNA is most exposed. And so we see this pattern on a gel. And what this is like this fragment is about 160 base pairs long. This is about 2 times 160 and 3 times 164 times. Because it's just randomly cutting by an endonuclease, so it's randomly producing pieces. that are most likely these little pieces that are covered by a nucleosome. And, you know, we can talk about that just because, you know, it fits with what we learned. But also, again, way back, scientists would run gels and they'd be like, you know, it looks like these cells are dying. And other scientists would be like, you want to fight, loser? I'll fight you. You know, you're a wuss. You're a scientist. And you're like, you're a scientist. No, you're a scientist, right? So they would run gels and they'd be like, well, what do you think is going on with this DNA, you know? Because it's doing that. That's just a hallmark feature of apoptosis. The DNA gets fragmented into these nucleosome-sized bites, right? So that's just that. So it's a little bit there for historical reason, but it's satisfying. It's another example. And then that goes to this, that in addition to activating the scramblase, we're going to activate that DNase, and we're going to activate it by destroying an inhibitor of it, and then it goes to town. Yep. So that inhibitor's there because it would be dumb to chew up your DNA, right? And you're like, I want to do something dumb. Okay, so that's the next one. And then we'll just talk about this one just a little bit, and that is there's also a really significant role for the mitochondria, and we'll talk about it. But off of this list from our book, we'll learn the big three, those big three. There are others, but these are really three satisfying stories, separate stories. And there's an activator, and the activator is a family of proteins called caspases. And remember, we use the term, and it's kind of a sloppy term, but we use the term effector. So these caspases are the effectors. They are the death protein. When they're activated, this cell is going to die. How's it going to die? It's going to flip its phosphatidylserine. It's going to chew up its own DNA. There's going to be mitochondrial involvement. There are other things, big things happening where that cell is going through apoptosis. So it's dying. There are specific mechanisms that get activated. They get activated by these caspases. And there are orthologs of caspases in every plant and animal. And they are the effectors. They're going to cause the programmed cell death. And what we'll learn is that upstream of this is regulation. You know, it's off. What we're going to do to activate it. We're going to turn it on. The analogy is caspase is like the gun, the actual shooting mechanism. And the upstream regulation is like, well, leave it unloaded, leave the safety on, lock it up in a gun cabinet, hide the bullets, hide the key, all kinds of regulation. Because you know what? These caspases are in every one of our cells right now. Just going like, well, you want me to send this thing through apoptosis? Because I'm ready. And that's just an indication in multicellular animals and plants of how careful the organism is in trying to keep the cells in line. Because if they get out of line, it's cancer. And that's why this chapter fits in here, is because we don't want cells out of control. We want to carefully stimulate growth and development for, you know, embryonic fetal development, child development, and exercising and stuff, growth, all kinds of growth, healing from wounds. We want to do that very carefully, but we also just want to be ready, that if that starts getting messed up, we'll just be like, boom, you know, we don't need you. You know, like that. So the caspases are always there, and they're being inhibited. Like they're like, nope, always there. It's just shocking to see that, right? All right, so let's have a little concept chat. Go. And try to meet somebody else, right? You're after spring break again. This is your week after spring break, and you had a party hoedown working spring break. And I'll just sit here and text with my friend Arlie.
So, also I thought he was really interesting to me. So, like, obviously, we've always got hypothesis. I'm a little bit confused on where the magnetized thing is coming from. Like, it's always there, and then the picture kind of made it look like it came in, and like maybe that was like constructed. It don't make sense.
Well, it reaches the outside, there's a smaller organelle called the
That like comes into it, and then it
the autosome is what actually
I thought the autosome was made from organelles already. This was really interesting because I have a feeling that this is kind of like the celiac disease, like when you're so sick of your body's like eating other things like enamel and stuff. Like I wonder if that's kind of like what's going on.
And then I think two other important things are the scramulase.
So is there a difference between scramblase and basis?
That is
So scramblase is a base?
the translocation of the phospho long word that I can't pronounce.
So he said, this is what I was confused about. So like a phosphate, I guess, is just what switches the normal phospholipids, but a scramblase is what flips.
I think it is flips them. When it's activated, it starts flipping, and then it can run
The phospholipid, the phospholipid theory.
yeah, so that's what flips it out, and that's what it's
Because you said that there was a specific base which is phospholipid to the outside. So is that specific name? Okay, I need to ask because I was so lost.
Okay, next one is inefficient mayonnaise. Basically, it's just, we learned from endosome before.
Yes, like with the different methods.
Yeah, so, and it's specifically that one.
So basically, what you're just gonna make random cuts in the nucleus, like between the nucleus.
I would, say not random because it is in between the nucleosomes, but I think some can access those very well. But that's what makes the ladder is that it's each one is basement. But I'm sure it's like that's like one basement, that's one nucleosome, that's two, that's three.
Oh, I get it. I wasn't getting what you were saying.
So if you give it like another hour, like 4 hours,
Do-do-do-do.
there'd be a lot lower.
That makes sense. And then obviously we have to have upstream regulation of that I really learned.
So we're doing this for another minute.
And that it's controlled by activates that we all have. Basically like the words of the cell. I was like picturing like low alignment, like all those
So Justin, what's one thing your group thought was important so far today?
The hyperphosphorylation concept for.
Yep.
I mean, that's the first time we encountered that.
Yep, yeah, that's awesome. So P53 gets phosphorylated to be activated, initially binds to targets that are going to block the cell cycle and say, wait, there's damage. And then it just continue, that check 2 just continues to phosphorylate it, gets hyperphosphorylated, and that causes a little different change in its shape. It now binds to additional targets, and those additional targets are going to be these regulators of caspases that are going to activate the caspase. So we hinted, there's a bunch of upstream regulating the caspases. Those proteins are always in our cell. They're constantly being inhibited, and PP3 activates targets that overcome that inhibition. Perfect, right? And just satisfying, right? Because, right, when, these examples can be nice. They're not oversimplifications, but they're selected to be kind of sweet examples where you go, ding, 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 ding. We've learned there's lots of application, integration, and context dependence. So things get really complicated, but we need to have a framework, of how to move forward and build our knowledge beyond our course. So that's great. Cam, what's a thing your group talked about?
We were talking about basically the same thing, but.
That's cool. That's what I would say every time in college. I'd be like, yeah, ditto, love you too, like that. Yeah, but yes?
But it was something a little bit different, so I was asking a question about the pathway that P53 goes through, and so it binds to P21, right, and that's the inhibitor?
Yep.
What is it inhibiting it from?
So that's a good question. So always, and sometimes you say that's a good question. They're always good questions, But so P53 is a transcription factor. So you can invoke your clay model videos from the first day of class, right? It's a transcription factor. Its job is to bind the enhancer of target genes. What are its target genes? And the answer is when P53 has one phosphate on it, its target genes are things like P21 gene. P21 codes for a protein, a small inhibitory protein. And so the P21 gene is there. When it gets activated, it starts transcribing into mRNA, and then the mRNA gets translated into protein, and it's this P21 protein, and it is just going to bind to CDKs and say, we're stopping the cell cycle no matter what else is going on. You're not going into S. It's the DNA damage checkpoint within the G1 to S transition. It's like that extra school patrol officer that doesn't, you know, everything else can be green and everybody can be waving the cars, go, go, go. But the school patrol, the P21 can just make it stop like that. And that just continues. Stop, stop, stop. Then, and there are other targets, but one target is enough. You know, there are additional targets. Then, as the DNA damage is trying to get corrected, if it doesn't get corrected quickly, the check 2 stays active. It's a kinase, and it just keeps adding phosphates to additional sites on p53. And when p53 gets hyperphosphorylated, it starts to bind to additional target genes. And that's a surprise, because we didn't hear that about MyoD or myogenin or ALK, you know, that they could shift. And so when p53 is hyperphosphorylated, it now binds to additional target sites. And it includes the activation of genes that are going to code for proteins that will stimulate the caspases.
Wait, can you go back a second? So it gets
Yep.
hyperphosphorylated by an excess amount of CDKs? Is that what that
No,
is?
it gets hyperphosphorylated by time waiting with the DNA damage not corrected. Because the kinase that is phosphorylating it is a kinase called CHEK2. And CHEK2 just keeps phosphorylating it because it's not, the DNA damage hasn't been corrected, so it just keeps phosphorylating it. And in this case, it starts phosphorylating other additional sites, and that shifts the shape of the p53 protein so that it now binds to additional enhancers on these other genes. So with just a first round of phosphorylation, p53 binds to a set of genes that include inhibitors of the cell cycle. Then as more phosphorylation occurs, it binds to another additional set of genes that are activators of caspases. Yep. Yeah, and it's like this little timer mechanism, you know, like that. Like, how do you build a little molecular clock? And you're like, well, there's one, you know? And you're like, geez, man. And like you asked, you know, could there be a variety of mutations in that system? Ah, yeah, right. P53 itself is a famous tumor suppressor, right? And you can think about that there would be some other genes in those pathways that would also be tumor suppressors. And if they got mutated, you'd be in trouble. And then there would be some that are in that pathway that end up actually being oncogenes, because if they got mutated, you wouldn't have that function anymore. Yep, and that gets complicated. Our book only goes there one time in chapter 20, that idea that, whoa, it's a pathway. So if you have one player in the pathway, it's a tumor suppressor, well, along the pathway of activating it, who works with it, who works opposing it, and those could be additional tumor suppressors or oncogenes. Yeah, so again, our book and our class will only go there one time, because it is a thing where like, ooh, yes. And I invented this recognition. Right, hold your hands like this. Everybody hold your hand like this. We'll do a little quick learning. All right, hold your hand like this. Don't change your fingers, but then hold your hand up to your face like this, right? That's one emotional state. You can think about, what are you expressing when you're thinking like that? It's kind of skepticism and it's kind of doubt, and you're like, I'm not going to talk, but I don't really believe what Dave's saying in class right now. And then there's this other one, and I claim this is a different thing, yeah. And maybe there'll be a little extra credit for the first time on any of my exams on the final. They'll be like, what do you think those two emotions are that you're trying to express when your hand's in the same position, and you're like, And I have a friend who does this almost constantly when I'm talking. They're just like, and then I'll be, uh-oh, you're like, whatever. Okay, sorry, Alex, wait, let me get in position. Alex, what is one thing your group talked about that you thought was important?
We were talking about how the outer leaflets can't move inside to the inner leaflet, and then we were talking about how the phosphatidylserines
Yep.
Can move to the outer leaflet because of the fluid.
Interesting. Yeah, that's another one, right? And those are just so satisfying. Well, I just love molecular biology. You know, when I ran into that in 10 minutes, I was like, I love this. Because it's like car mechanics. there isn't any gas in the tank. What do you think's going to happen when we try to start the car, right? It is that, you know, it breaks it down, and it's all at this molecular level, so it's way below the ability to really visualize it in any kind of microscope. But it's like you can set up experiments, and you're just like, what do you think's going on? So yeah, absolutely, yes. Questions?
I have a question. So what's the difference between flip base and the scramble base?
So flip base, my way of understanding it is flip base is this broad category, a term for proteins that can flip phospholipids from one side to the other.
Okay.
And then there's a whole bunch of them, and they'll have specific names. And sometimes the specific name people like to use is flip base for that specific one. So it's frustrating in teaching and learning the topic. Because we learned the word scramblase. But scramblase is also a general term. Like that. And I can go into more detail about, well, I think there's a way that the general terms are used in a specific sense within the field. But it's not clear to me that everybody's adhering to that general rule. So it's a frustrating thing to teach and learn because it's something that's simple. It's just there's a lot of them.
Is there a term you prefer us to use?
Is there a term for us to use? Scramblase. Yeah, we're saying that's the specific protein. There's a specific protein that flips it, and we're just calling it scramblase. My understanding is that phospholipids are synthesized in the smooth endoplasmic reticulum, and they're only synthesized in the outer, what is eventually going to be the inner leaflet. And then in the smooth endoplasmic reticulum, some of them, as soon as they get synthesized, get flipped to the outer, because they only get synthesized in the inner leaflet. Because that's the side that's facing the cytoplasm, and that's where the enzymes are that are going to do the synthetic pathway. Yep. Questions people have? All right, so we're doing great. We have like 2 more minutes. We talked to some people, and I got a message from Arlie. No, I won't look at it right now. Let's move on. So other questions people have about anything at all? Yes?
So based on that, a scramblase is technically a flip base. It's a specific.
Yeah, for our kind of course, we would say the broad term is flip base, and we're using scramblase as a specific example of a flip base. Yep. Yeah. Again, the terminology within the field, you get the hint that they're using it specifically sometimes, but then you hear somebody who's in the field go, oh, well, they're all scramblases. And you're like, that's not helpful. Yeah, so it's a little bit frustrating, that terminology. You know, I'd like it to be, they're nice, they're consistent, the term is consistent with the kind of stuff they do, but it's just a kind of sloppy, frustrating, yep, terms. Yep, yeah, but again, yeah, good question. For us, the specific one, we just call it scramblase. And then it's an example of a broader category of proteins that flip bases, like that, yep, yeah. And there are lots of them. Sam mentioned flop bases. It just, you know, it's just, I think the terminology is sloppy. As far as I can understand, I'm not a super expert in that, you know, I don't research that topic, so I just see it from outside and going like, wow, you guys clean this up some. Yeah, and scramblase is certainly what our book uses too, so we're going with that too, you know, like that. Yep, other questions people have about anything at all? So let me just scroll back here as we finish our class and you start to pack up, because that's a nice list to start doing this thing, right? We're going to roll through this chapter pretty quickly. We'll certainly finish talking about it on Tuesday, and that's because you have this stem cell assignment that will come separately like that. So read the whole chapter and then go on to start reading chapter 19, because we're going to test 2 weeks from today. Awesome. Oh, yes. And if you have a homework you want us to hand in, the cell cycle stuff you want to hand in, great.
My corrections.
Oh, good.
And I just wanted to, so I don't know if you know, I have C. diff. I just found out like right before break. It's like an infection caused by antibiotics. I've been really, really sick.
Yeah. Oh, you sent me an email already?
No, I hadn't sent one.
Oh,
I've
yeah.
been like knocked. I was in the hospital.
Oh, my God.
I am hoping that like I get better, but I'm just like warning you in case we need to have like a later discussion.
Yeah.
I've been like really
Yeah,
sick.
yeah. So our course was built this way long before COVID and the pandemic hit.
Yeah.
And when that came out, I mean, I was horrible.
Yeah.
But for teaching, I just
Yeah,
right?
so having the
because
purpose.
yeah,
Mhm.
because the goal is to be able to keep on learning
Yeah.
like that. But it's a big deal that just don't worry
Yeah,
about it, because
okay.
our stuff is here and you'll be able to complete this course
Okay.
fine. The lab course is different because that's more like
Yeah.
participate, stuff like that. So we have to talk about so
Yeah.
don't worry about this course. You know,
Okay.
if there's delays, we'll
Yeah, I've
work
been
it out.
keeping up with like,
Yep.
the work and the
Yep.
videos and stuff.
Yep.
Okay.
Cool.
Alright, thank you.
Hi. Oh yeah? Yeah. You
Is
said
there more information on when we're going to be presenting at Thinking Matters? Is that all? Are we going to go over all
The time of day?
the time of day and where and what we like, who I send my PowerPoints to, and
Yeah, there will be. So you submitted yours, and you probably got a response saying you
No.
received it.
oh, I got a response saying they received it,
Yeah,
but nothing else.
that's fine. Yeah, and so I'll just tell you from previous years, right?
Mhm.
Like that there are some poster sections, there's some talk sections. I think there'll be like 4 chunks of like talk sessions, so early morning, mid morning, early afternoon, later afternoon, and in the past, last year anyway, it all happened at Abramson Center, which is the one that's on the other side of the walkway,
Okay.
like that, and everything's happening in there. And then they try to organize it by topics. So in general, they'll put MHR students together. And sometimes somebody like Michaela Reagan will be the moderator.
Oh, that'd be cool.
And then sometimes, the moderator runs that session, and sometimes the moderator says, we'll have these like 4 student talks in a row, and then we'll ask questions of any of the speakers.
Sure.
And other moderators will say, we'll have your questions after each talk. You know,
Okay.
like that? Because it seems to be that, you know, the moderator runs the session.
Yeah, yeah.
But
Okay.
it tends to be like that, and it tends to be like, you know, the MHR students are kind of clustered together.
All right, and
Yeah.
someone will get emails about this and.
Yeah, you could email them now, and you could just, you know, feign some,
Hey, I'm curious.
well, I hope I get placed in the morning. If you have some preference,
Sure.
you know, you
Yeah,
can
yeah.
say that, because the MHR students will be in a couple of different sessions like that. And so, you know, it's not like, you know, like like you say, if you had an appointment, you could contact them and say, I really would like this way.
Sure.
Or you could just be like, I'm leaving town, right,
Sure.
and I need to get out of there
Sure.
at lunchtime.
Sure. Follow up. I got an email from Mackenzie about a meeting on Monday
Yeah.
about
Presentations on presentations. Yep.
What is?
So that's like coaching to try to help students do a good job with presentations, and
Okay.
MHR is very quite anxious about that, just because it's a public-facing
Sure.
opportunity for them, you know, and they want their students
To look good
do a
and
good
feel
job.
good, yeah.
But not to pressure them, but just to try to provide support.
Yeah.
You know, for them to, you know, to say, oh, okay, you're feeling comfortable, like that, you know?
Okay.
And so, especially Lucy has a set of PowerPoint slides that are kind of generic that she goes through and says, you know, well, don't put color on them and don't make many slides. So it's about specific for, you know, formatting and planning the talk, and then to make sure, you know, you're communicating with your host lab, you know. Hopefully having a little practice talk with somebody there so they can be like, yeah, that's, you know, good. And also like how to properly like cite, you know, somebody like, you know, like I'm showing you this Western blot was actually done by Jim Dandy in the lab, not me, right?
That is important information.
Yeah, so like that.
Okay.
Yep. And it's all about, it's just focused on that. Yep.
Great.
100%. Yep. Oh.
Sometimes
Stop recording.